Hi, I'm Susan Ruediger, and I'm the co-founder and CEO of the CMT Research Foundation. Today, I'll be talking with Dr. Peter Marks from the FDA. He is the director of CBER, the Center for Biologics Evaluation and Research, and his center is responsible for the evaluation and approval of gene therapies. Dr. Marks and I are going to talk about the relevance of gene therapies, how they're important to people living with CMT, and what they could mean as a treatment option. We'll explore designing clinical trials. We'll explore challenges that are found within somewhat rare patient communities, and we'll explore how maybe gene therapies can be used for not only one type of CMT, but all types of CMT. I hope you'll enjoy the conversation. Thank you for joining me today, Dr. Marks.
Thank you for having me.
It's such a pleasure. I mean, I know in the time of COVID, your department is responsible for the approval of vaccines. And I know that your world is moving very quickly today. I'm really, really grateful for you taking this time. Thank you. So because you're responsible for biologics, that includes vaccines, that includes gene therapy, and that includes cell therapy, your division is going to be really important to the approval of drugs for CMT, especially anything around the world of gene therapies. And as you may or may not know, CMT is a genetic disease. There are over 100 different genes that are known to cause CMT, and it affects 1 in 2,500 people. That's 2.8 million people around the world, with 100 different genes and possibly even thousands of different mutations yet to be identified. We're looking at gene therapy as being a huge opportunity for us as a treatment option. But right now, quite frankly, there are no treatment options for people with CMT. And the goal of today is for us to talk a little bit about the FDA approval process, how patients can expedite that, how we can help you help us, and how we can really be partners in this process.
Sounds wonderful.
Can you tell me a little bit about the FDA and a little bit about your center?
So the Center for Biologics Evaluation and Research is the part of FDA that deals with the more complex biologic products. We regulate blood products, derivatives from blood. We regulate allergenic products. We regulate vaccines. We regulate cellular products, whether they come from humans or from animals, if they're given to humans. And we regulate gene therapies. And that includes the whole gamut of gene therapy, from doing gene therapy in cells to doing gene therapy in humans.
Excellent. And I know that that's a real hot topic right now in the world for rare diseases and rare genetic diseases. So at the FDA, what would you say your top priorities are for approving drugs?
So I would be, right now, I would say that obviously in the time of COVID-19, if I didn't mention that for our center, making sure that we have a vaccine for COVID-19, that would be pretty crazy. So obviously that's a top priority, but we have had a very long-standing commitment, and this dates back to long before COVID. It dates back several years to really trying to advance gene therapy, particularly gene therapy for relatively uncommon disorders. And although in aggregate, charcot-marie-tooth is, as you said, that is something I knew about it. Last count I had was 80 different genes, but now I know it's more than 100 genes, so you've updated me some. And although there's perhaps one particular gene that may be able to get some percentage, it's going to be a lot of individualized gene therapy. And we've been advocating over the past years for evolving a structure that is very conducive to developing gene therapies that are more individualized. We've worked, in fact, with the National Center for Advancing Translational Sciences, which is a nice connection to Chris Austin, who's the head of that, who directs that, to try to put together a program that it's called the Bespoke Gene Therapy Consortium, to work towards helping to bring forward gene therapies for small populations. Because when you have a disease like charcot-marie-tooth, which is really, it's really a conglomerate of these 100 plus other genes that cause it. So it's really 100 different diseases from the genetic standpoint, and you're going to have really individualized gene therapies. Yes, some will be for a larger group of those patients, the CMT1 patients, but some, even there, it's going to have multiple different gene therapies potentially. And so I think this is a really exciting time, because not only do we have the ability to start to address CMT by gene therapy, but we're trying to also have the pathways so that we'll be able to make it so that after the first advance for a group of patients comes through, we're able to follow that up relatively quickly, hopefully, with things in its wake that will come forward more quickly because of that first approval.
Yeah, you know, we've heard that a lot, that we need to have one good treatment approved to really start the wave and start the momentum. How many gene therapies and approvals do you have right now?
So right now in the United States, there are 5 approved therapies. Now that doesn't sound like a lot, but and 3 of them are CAR T cells for cancers. One of them is a gene therapy that's given directly into the eye for a type of hereditary blindness. And one is, as you've mentioned, for spinal muscular atrophy. But that doesn't take into account the fact that we have over 1,000 active investigational new drug applications. Those are what has to be submitted to us before you give an investigational treatment to humans. So those are not approvals. Those are things that are in clinical studies. But that's an awful lot of things being studied in terms of gene therapy. So there's a lot of work in this area and a lot of excitement. A lot of it is using a particular gene therapy vector called adeno-associated virus. And that's helped us to make a lot of advances. And we're trying to continue to build upon those advances as we move forward here.
Yeah, you know, the CMT Research Foundation is funding a project right now for CMT1A using AAV as a delivery mechanism. And we're 18 months into a 2-month project as of today. And we're really excited to see the progress that it makes, and hopefully to be able to partner with you all to usher it along. So can we talk about that a little bit? Talk about partnering with patients. I mean, I think a question I get a lot from the patient community is, what can I expect a gene therapy will do for me? And how will I know whether I want to take it or not? How do you all partner with patients to understand the disease, to understand the urgency, to understand how desperate of a community we are, and how much we want to drive this forward, but yet how important safety and efficacy, and how do we marry all of this together?
Well, from our center's perspective, one of the ways we do that is through patient-focused meetings, where we have people that will meet with a patient group to understand what their concerns are, understand what worries them about a potential therapy for gene therapy, a potential gene therapy, what makes them concerned, what they feel is most important about in addressing their disease. Because sometimes what doctors think is the most important thing is not what's most important to the patients. And we need to understand that. So that's one of the things we can do at those meetings. The other thing that we can do at those meetings is help patients organize, because sometimes one of the key things that we need at FDA is to understand how people document how they're functioning before they get some gene therapy. Because the way we understand what a gene therapy really does is by comparing it to some baseline, or by comparing it to people who don't get the gene therapy. Now, in a very small patient populations, doing these comparative studies, also known as randomized controlled trials, they can be tough because people don't like getting an inert medicine called a placebo. They would prefer to get the gene therapy. One of the ways to do that in gene therapy, because some gene therapies really do bring significant benefit, is to understand the natural history of a disease, and then to show that after you've given the gene therapy, you've made a very distinct change in the the natural history of that. It all depends. Some diseases, it's easier to actually do a randomized trial because it's hard over a period of 6 or 12 months to see differences in a patient from how they were before they got the gene therapy to after. In other diseases, it's easy to do that because, for instance, in spinal muscular atrophy, that was a good example. There, if you don't give the gene therapy to a child by the time they're 6 months of age, in spinal muscular atrophy type 1, by the time they are 2 years of age, they're limp and on a ventilator. And so there you knew by intervening, you didn't need a control group because you knew the natural history. Mm-hmm. And so these natural history studies are something we encourage patients to do, because if you can find some measure that changes relatively rapidly, or that can be measured relatively well over time, it allows one to see what the effect of a gene therapy, potentially without needing that control arm that's done at the same time with randomization. So they can both things can be done. And one of the things we do sometimes do is have trials where the two groups, the group that gets the gene therapy and the group that gets the control arm, after a period of time, the placebo arm crosses over to get the active gene therapy. So that can be done as well. But understanding the disease, to get back to your question, is really helpful for us. Understanding what matters for patients, and then something that patients can do for themselves and you can do together is really help us understand what's the course of your disease. And in some cases, FDA, the larger FDA, the Orphan Products Division actually has grants that they give out so that patient groups can get funding to put together registries or help understand what the natural history of disease is. So we very much support that. And it can make a big difference in helping developers along in their process, because they are focused on the actual gene therapy. They're often not focused on the kinds of evaluations that are needed to show that therapy actually works.
Yeah, so the drug development and approval process is really a partnership between patients, clinicians, the drug developers, and then the FDA and the regulators.
That's absolutely correct. It's everyone has to work together to do their part in this. And we're grateful for patients for participating in trials. And we're grateful for the input that they give. And it's very clear, I think, from some good case studies, that patient groups that have worked to organize and have done a really good job in helping us to understand what really matters to them, what the course of their disease is, that actually makes a difference, and it helps the product developers as they move forward. Think about that, an example of a large group that's done that is Cystic Fibrosis Foundation, where they've done a really great job there, and they've even helped fund a lot of the work that's been done, but it's made a big difference. Same thing can happen on a more micro scale, and although CMT at 1 in 2,500, it's not super common to people perhaps, but it's also not super rare either. So and so it's important to get this right.
So let's talk a little bit about, the approval process and clinical trials for a quasi-rare disease. And of course, within CMT, I think we need to look at, especially in the world of gene therapies, that instead of being one disease of CMT, that we're actually looking at hundreds of diseases, because each one will have its unique gene therapy. So can you talk a little bit about a clinical trial for a small population and what kinds of programs the FDA has to address the unique concerns of rare, ultra rare populations?
Right. So I think it starts by having us, when we have oftentimes these diseases will be addressed at first by an academic group, and we have early meetings that we're willing to have with folks even before they start the official regulatory process. We call them interact meetings. They're very early meetings where we don't, they're not formal meetings. They're kind of informal meetings where a sponsor tells us, okay, we'd like to develop an AV therapy for CMT1 or one of the CMT types. This is what we're thinking of doing. Here are our questions. You know, how many patients will we need to study? How do we have to do the manufacturing of the product? You know, and things like that. So those meetings can help to set up this process and set up how we set up the sponsor for success as they move forward. Then after that, they'd submit an application, an investigation of drug application, which we then more formally review and get back to them with any questions that they have in responses to those. And then ultimately, there will be an IND application that we allow to proceed into trials. For rare diseases, we're increasingly hoping that, I would hope that where we're headed here is for CMT, that you'd have the large indication, the largest of these, which would pave the way for subsequent AV therapies. So one thought here that we've been exploring, and it's not fully baked, but I'm willing to share it with you, is this concept that the first gene therapy that comes along for the largest population would use the endpoints, they would help trailblaze the endpoints that would be used for subsequent trials. It would actually also trailblaze the manufacturing process, because the idea here is that hopefully what we'll find is that we could use the same vector, which is the vector is what gets the gene therapy in. And it can be, the best way to think of it is it's like a razor, okay? You just reuse it. You can reuse it over and over again, but you change the razor blade. And the razor blade would be the gene therapy construct that addresses each of the different CMTs, the 100 plus different gene errors. Now, it may be that for CMT, because there are some things or duplications, et cetera, it may be that it'll turn out that another type of gene therapy construct called genome editing may be more advantageous, but let's leave that behind for today. But the idea here is that for at least for some of these, one could use this razor blade analogy, and the first one you'd need to do a lot of work on in terms of characterizing the gene therapy product, and the clinical trial, though, we try to streamline as much as we could to get the answer in with as few patients as we could. But once that's done, the idea would be the next one that comes along leverages the manufacturing information for the vector. It leverages the clinical trial endpoints used for the first one, because again, the outcome measures may look very similar for a number of different CMT variants. So this should help streamline getting through the 100 different mutations. Again, as I said, I want to be careful to say not everyone may be addressable by AAV, but there are other gene therapy techniques that are under study right now that may address those others. One of them is this genome editing, where you can fix either individual mistakes in the genetic code, or you can actually just chop out pieces of DNA if you had to.
I have a really big question. I have CMT1A, and my question is, let's talk about gene editing for a non-fatal disease. I mean, CMT isn't usually fatal. It can be sometimes, but not usually. And it certainly isn't fatal in early life most of the time. Certainly it has been, but usually it's not. What is the FDA's appetite for gene editing or any gene therapy for a seemingly moderate to moderately severe disease?
You know, I think we look at each case individually. I think that for genome editing, it's actually not a crazy thing to talk about when we have genome editing advanced to a point that we feel like it can be done safely. Because what we're talking about for CMT is we're not talking about fixing at least at this point in time, we're not talking about fixing your genomes for your offspring. That would be heritable genetic modification. That's something that that the National Academy of Medicine and their international bodies have said it kind of off limits right now for truly non-lethal diseases. And there's even for lethal diseases, there's still tremendous controversy for heritable genetic modifications. On the other hand, for something like CMT, where once we have a better sense of how these genome editors can work in the body, it's not a crazy thing to address. The major problem that we have to deal with right now is we have to understand how much fidelity they can have for just correcting a given mistake. The problem is that when you go to correct one mistake, you don't want to create many others. And so that's the problem with some of the genome editing technology right now. We're not sure when we go to correct that one mistake, whether we're just going to, we don't want to make things worse than they could be. We might correct that mistake, but if at the end of the day, we make some other mistake that causes the person to get a cancer or to have some enzyme stop working that they need otherwise, that would be bad. So we're certainly working on getting those data. And I don't think we're that far off. I mean, I think this is something that in the next few years we'll learn more and more about, because people are doing the first steps for genome editing in the kind of a safe harbor of doing them in cells that are given to people. So by doing that, you at least can control where the editing's done, and you know that if there's been some mistake, where it's confined to. At some point, we'll get enough courage, instead of just doing it in cells, to be able to give it to somebody in their vein and know that we'll feel confident enough, but we're not quite there yet.
Okay. You know, it's a question that's at the top of our head at the CMT Research Foundation, and it's at the top of my head as a patient, to think about editing genes to stop my neuropathy, or maybe even potentially reversing some of my neuropathy. And it's exciting for us, and it certainly is exciting to think about the prospect of doing something like that for a young child who has no symptoms and preventing the symptoms from ever happening. It's an exciting proposition, but certainly one that is not to be taken lightly. And we think that our job at the Research Foundation really is to create as many opportunities as possible, partner with industry who wants to carry it forward, and then convince the market that this is something safe and effective and worthwhile and that it reduces our disease burden enough to be worth it. Yeah.
Is that because one can use certain you can basically use electrical impulses to potentially, to help understand what's going on here. You potentially don't have to just rely solely on clinical endpoints. So that may be helpful here as well. Right. And I think the point being that I'm trying to make is that because you'd like to find, if you can, things that will allow us to do clinical trials in a shorter period of time. And sometimes if you can find that type of marker that's a physiology marker, even though ultimately what matters is how patients feel, function, or survive. Okay, that's a given. But sometimes if we can connect that, if we can use the marker, we can sometimes get there more quickly than doing a multi-year study in order to get to that clinical endpoint.
And in a disease like CMT 1A, for example, that's a generally slowly progressive disease. I don't want to be in a clinical trial for 5 years, and we know our industry partner's not going to want that either, and you're not going to want that. So we want to find those pieces of information that give us positive results fast.
Yeah, that it's not advantageous to have clinical trials go on for years and years, in part because the danger when one has a clinical trial that goes on more than a couple of years is that there are changes in the standard of care. The rest of the world is changing, so that sometimes then by the time you get to the end, you're back at the start, because things have changed so much that you can't really use the data that you've found.
Right. Yeah, that makes a ton of sense. So, is there anything that patients can do, in your opinion, to expedite the drug development process or the drug approval process in this case?
Yeah, well, I think the thing that patients can do that most helps us is by participating, helping to make sure that we have the right natural history studies, that we really can understand. The more we understand the disease, the more we can help sponsors understand what the best endpoints would be. And so I think coming out of this, one of the things that I can say is that a continued dialogue with the agency, perhaps, around what we can do working together, patient focused, is certainly worthwhile, because I think that helps us, understand. It helps us to help the sponsors make realistic expectations for their trials.
Yeah, and I think that's one of the things that a patient foundation can do, and I'd love your input on this, but certainly something that I'm doing in my role is I'm a patient living with this every day. I actually come from 6 generations of people who have
Wow.
this disease. So I can sort of predict what's going to happen to me when I'm 75 by looking at my mom and my grandmother and et cetera. But, we can connect the researchers, the academics, to the industry partners, to the regulatory partners, to NCATs and the government partners, and the patients, to bring them all together. We like to call ourselves the shepherd of the research, and connect, being the great connector of all the key players. That's a job I've had a lot of fun doing. But then also as a patient, having my voice be heard by people like you and our pharma partners and Dr. Austin, of course.
Yeah. No, it's so important. I mean, I think this is people feeling empowered to connect with us is really important. Um, I think what you're doing is very important, because making that connection will help advance the science so much more quickly than if it's just passive. And unfortunately, what happens in rare diseases is the squeaky wheel does get the grease to an extent. And it's good to be proactive, and working towards this. As I say, I think this goes back to what can you do? I think helping us understand the disease as best as we can, what would matter the most, helping to identify populations. It may be that a certain age population would be the best time to, sometimes if you study something in the entire population, it may be very hard to see a change. On the other hand, if you study a certain age population, you might be able to see the change more easily. So helping us to figure that out is incredibly useful. We benefit from it. I think the patients benefit from it, and ultimately that makes progress happen the most quickly.
Well, thank you. I mean, please know that I'm here to be an active partner with you in this process.
We do expanded access for products that have shown there are various methods of expanded access. There's single patient expanded access, which is done when you have one individual with a severe disease who you basically are trying to get a therapy to where it could be potentially life-saving. Then there's expanded access for intermediate treatment size populations, which is probably what might be applicable here, so that if one saw that a gene therapy seemed to be helping people in a clinical trial, you could potentially make it available to those who don't exactly qualify for the clinical trial as part of expanded access.
Okay.
I should be completely transparent and say that to date, for gene therapy, expanded access has been a little challenging because of the cost of the gene therapies to produce. And so, especially because as you start to think of treating larger people, as in anything larger than 20 or 30 kilogram child, the amount of gene therapy that you have to give if you're giving it systemically is significant. And so it gets to be somewhat costly to produce. Nonetheless, we're very amenable to expanded access. My hope, though, is that and I think this is shared, I don't want to speak for him, but I think it's shared for some of the folks at NCATS, is that we can find a pathway forward that allows us to do clinical trials that are inclusive enough that we're able to bring in enough patients into those trials and learn from the patients in the clinical trials so that every patient that goes through helps contribute to that. Because ultimately, you don't want to make a rare disease even more rare by saying, well, we'll take CMT patients between X and X age, and they have to have this much or this. Obviously, we'd like to have the most inclusive trials. One of the ways that sometimes can be done is a little bit of a trick in rare diseases is one can have two populations that run, that are enrolled into a clinical trial. The kind of the primary analysis population, which can be very defined so that those are the patients that you think you're going to be able to see the clinical endpoint in more easily, and you power the statistics for that population. But then you have an additional arm where those are patients who may not meet all those criteria, but you're at least able to get safety data and perhaps some efficacy data, but it's not going to be critical to the analysis. So we'd like in rare diseases to get as much information from every patient that participates. And that's not to say that expanded access is bad at all. It's just to say that in rare diseases, the more information you can get as part of a clinical trial that contributes to ultimately getting across the finish line to an approved product, the better.
I think it's really important that the patients enroll in the clinical trial database and visit a CMT clinical care center where they can be evaluated by an expert who's involved in this consortium of doctors who are evaluating this together. And we certainly know that patients need to be the squeaky wheel, and by partnering with the Research Foundation, they can do that to start squeaking with you all. When you take a look at the landscape for CMT, and I asked this of Chris Austin and a few others, what excites you the most about treatments for CMT? I mean, here we are with zero in September of 2020. What excites you the most?
I think what excites me the most would be that right now there are a variety of, I'm aware of a variety of clinical trials of things that probably will be modest improvements, whereas a gene therapy could potentially be transformational. And I think the idea here would be, I think if one could make inroads with a first gene therapy, this is a great paradigm for how one could come along and use this razor blades model to try to essentially bootstrap through a lot of different underlying genetic causes of what looks outwardly like the same disease. So I think this is it's an exciting time because as opposed to making small incremental progress with a drug, a gene therapy could make tremendous progress by actually fixing the problem. Would it be for life? We don't know that yet, but it could be. And so it's just an exciting time to see gene therapy moving forward. And I think this is a great example of one of the conditions where it could be applied and make a big difference.
Thank you for that, because the CMT community has had 30 plus years of no hope. The disease was named in 1886 and the first gene was found, CMT1A was found in 1990, published in 91, but actually found in 90. And so we've gone 30 years with knowing what the cause was of the most common type and yet not having any potential therapies. And hearing your excitement and your enthusiasm, I think that will be hopeful for the patient community. And of course, it's important that we go through the right process to make sure that it's safe, to make sure that it's effective, to make sure that we're doing this the right way. But I'm grateful to see your enthusiasm. I certainly feel it, and I'm glad it's shared by you.
Yeah, and I think our folks back at FDA who work in this area of gene therapy, as well as the people who work in patient-focused drug development, we're all excited about this too.
That's great. That's great. Well, thank you for your time. I really appreciate it.
No, my pleasure. Thank you. It's been a pleasure, and I hope we get a chance to meet at some point.
The FDA is a key partner in the development of treatments and cures for CMT. They are determined to ensure that treatments are safe, effective, and address the patient's most urgent needs. Partnering with patients, key researchers, and drug developers to fully understand a disease like CMT is just paramount to success. We really look forward to working with Dr. Marks and the FDA in the near future as we're evaluating potential treatments for a disease which right now has zero available. I know the CMT community, we're really eager to have treatments developed and approved and available for patients. And we really appreciate Dr. Marks and the FDA's commitment to safety and efficacy and his partnership in the drug approval process.